Good morning. My sincere greetings to all colleagues and friends everywhere. My talk is direct binding of a small heated shock protein αB-Crystallin and spike protein of SARS-CoV-2, coarse-grained molecular dynamic simulation. Almost 2 years ago, mankind was plagued by a highly infectious disease called COVID-19. WHO confirms the infections have become 225 million cases. The main symptoms or the common symptoms of this disease include headache, loss of smell, and nasal congestion, and runny nose, cough, muscle pain, sore throat, fever, diarrhea, and breathing difficulties. SARS-CoV-2 is an enveloped, non-segmented, positive single-stranded RNA virus. This virus has 30 kilobase per genome. That genome encodes 14 open reading frame genes. Open reading frame 1A and open reading frame 1AB genes encodes polyproteins, about 16 non-structural proteins by autolytic process. Recently, affinity purification combined with mass spectrometry approach uncovered there 332 protein interactions between SARS-CoV-2 and human proteins. The interaction between SARS-CoV-2 proteins and the human proteins highlight how many protein complexes and biological processes can be influenced. The total number of SARS-CoV-2 proteins about 29 proteins. SARS-CoV-2 proteins are localized into different cellular compartments and subcompartments inside the cell that provides valuable information about its function and possible interaction with other proteins. Craig reported temporal alteration in host protein thermostability during infection, which converged on cellular processes including cell cycle, microtubule, and RNA splicing. 40% of SARS-CoV-2 interacting proteins are associated with endomembrane compartments or vesicle trafficking pathways. Angiotensin converting enzyme 2 is widely expressed in cardiomyocytes, cardiofibroblasts, and coronary endothelial cells, which are also a regulator for heart function. It was highly expressed in pericytes of adult human hearts, which indicate an intrinsic susceptibility of heart to SARS-CoV-2. Pericytes are blood vessels cells. The availability of αB-Crystallin for its usual function may be impaired and contribute to symptoms develop. Deficiency of αB-Crystallin augments ER stress and induces apoptosis by enhancing mitochondrial dysfunction. The alpha crystallines share sequence and function similarity with small heat shock. Now, materials and methods. Initial configurations for the biochemical or for biomolecules studied were obtained from the protein data bank. The NMR structure of the alpha-B-crystalline domain dimer was 2 KLR. Non-glycosylated receptor binding domain of SARS-CoV-2 spike protein and 2 KLR were docked employing CoDock-PP engine implemented in a molecular docking online tool, COVID-19 docking server. The best energetic pose conformation of the docked alpha-B-crystalline domain dimer SARS-CoV-2 spike RBD protein RBD protein complex was selected. All simulations were performed with the program package Gromix 2019.5 using the version of Martini 2.2 within the default time step 20 femtoseconds. The size of the cubic throughout the simulations was 12 nanometers. The non-bonded interactions between the coarse-grained solvent and solute particles were modeled by truncated and shifted Lennard-Jones pair potential with a cutoff radius of 1.2 nanometers. Water was also represented by coarse-grained water model. In this model, one single Martini water model represents 4 water molecules. 0.15 molar sodium chloride ions were added into the system to maintain physiological ionic strength in addition to the charge balancing counter ions. Trajectory analysis was carried out for 600 nanoseconds from each molecular dynamic simulation using the Gromix analysis tools. Figure 1 shows the This is the residues involved in formation of hydrogen bonds and the distances between these residues. And this sum about 44 bonds formed, hydrophobic bonds formed between the two proteins, and these are some, for example, and the distances of these bonds or the length of these bonds. Here we can see root mean square division we can observe that the wild type system equilibrated after approximately 75 nanoseconds. However, fluctuations were visible over the course of simulation, with the structure maintaining at a level of 1.5 angstrom after 600 nanoseconds. The root mean square simulation showed that the bound protein of αB-Crystallin involved in formation of the complex maintained an overstability throughout 600 nanoseconds of simulation, while the wild type displayed more fluctuation. The comparison of the backbone of spike protein in the bound state and the native one show that both systems of the protein stayed folded with a stable structure during the explored time scale, and this was less than 1.5 nanometer. The fluctuation is less than 1.5 nanometer. Here this figure shows the root mean square fluctuation, which express the mobility of the residues of each residue in the native protein or the protein involved in the formation of the complex. As we can see, the first 30 proteins show decrease in the mobility, in the dimer involved in complex. However, some residues in the dimer complex represent the binding domain formation in the residues 123 to 128 show some higher mobility than the native structure. In COVID-19, the mobility of the residues in the spike protein shows decrease in the dynamics relative as compared to the native structure, except at the region of loop 1 in the spike protein. Here is the center of masses. If the two center of masses have the same distance along the simulation experiment, this means that the two proteins are still in the same binding state. And as we can see, all over the simulation in these trajectories, the distance about 0.5 nanometer. This figure represents the radius of gyration, which is a measure of the compactness of a protein. This means no unfolding was happened. The radius of gyration of complex gradually decreases until the structure reaches a stable position 2.1 here. The radius of gyration of αB-Crystallin dimer started at about 2.3 and gradually decreased at 266 nanosecond to 1.5, and then level off. We can see here the solvent accessible surface area. All the structures have no change of the area between the complex, the two proteins forming the complex, and show high stability of the complex. And this figure shows the interaction between in yellow color spike protein and in green and light blue the dimer of αB-Crystallin. Here at 0 nanosecond, after 150 nanoseconds, the interaction become more clear, and 300 nanosecond the interaction increases, and after 600 nanosecond we can see that almost the two subunits of αB-Crystallin make interaction with the spike protein. Thank you.